Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so think about this for a second.
Okay.
Somewhere between 20 and 30% of men experience premature ejaculation.
Wow.
That's a lot of people.
Right.
And it's something so common, but what's actually happening in the brain
Right.
during, such a fundamental process has been a bit of a mystery.
Yeah.
Until now,
Yeah.
scientists are taking a really deep look inside the brain
Right.
to figure out basically the mechanics of how sexual behavior unfolds.
Mm-hmm.
And this could lead to some really surprising new ways to address
Absolutely.
these issues.
Yeah. What's really fascinating about this new research is that it's not just about the beginning of sexual interest.
Right.
It's about tracking the entire sequence
Yeah.
from that initial interest all the way through to the climax.
Right.
That complete picture has been missing for a long time.
Exactly. And that's exactly what we're going to be diving into today.
Yes.
We're going to unpack a recent study, a really detailed study
Yeah.
that followed the brain activity in male mice through the complete sexual behavior process.
Interesting.
So imagine getting like a front row seat to the brain's control center
Yeah.
during this entire experience.
Cool.
Yeah.
So if we think about why this even matters, to you, the listener,
Yeah.
understanding these basic brain processes could actually open up potential new ways to help with different kinds of sexual difficulties.
Absolutely.
So it's not just about scientific curiosity. There's a real possibility for new treatments down the line.
Yeah. And our guide on this journey is a very thorough scientific paper published in the journal Neuron.
Okay.
And we'll also be looking at a related article from a health news outlet,
Great.
just to kind of give us a broader perspective on the research.
Yep.
These sources give us a very detailed look at the brain activity involved.
So for you, our listener, our goal is to really pull out the most important insights, those aha moments where you understand the brain chemistry behind these complex actions.
Exactly.
We want to make it clear and interesting, focusing on the key players.
Right. The brain chemicals involved and how they all work together.
Yeah.
Without getting bogged down in, overly scientific language.
For sure. So let's get into the study itself.
Okay.
The researchers had a very specific question that they wanted to answer.
Right.
And it wasn't just about seeing if brain activity changed during sex,
Right.
but really pinpointing exactly how it changed
Got it.
during the different stages.
Okay.
So they were particularly interested in what happens in the brain as the mice transition between mounting,
Okay.
then insertion,
Uh-huh.
and finally ejaculation.
That's a crucial distinction.
Yeah.
They weren't just looking at whether the on switch for sexual behavior was flipped.
Right.
They wanted to understand the specific steps the brain takes
Right.
during each part of the process.
Right.
That level of detail is what makes this study so valuable.
And to get this detailed view, they focused on a very specific brain area
Okay.
called the nucleus accumbens.
Yeah. Now, this area is often talked about as being really important for pleasure and reward.
Right.
It's a central part of the brain's reward system.
It is.
So think of it as a key hub for those good feelings.
So what's particularly interesting here is that while we know the nucleus accumbens is involved in feeling good, being motivated,
Yeah.
this study really looks at how it contributes to the sequence
Right.
of sexual behaviors,
Yeah.
which hasn't been fully understood before.
So to actually watch what was happening in this area,
Yeah.
the researchers used male mice,
Okay.
and they actually injected tiny sensors that glowed under a certain light into their nucleus accumbens.
Wow.
And these glowing sensors were designed to detect the release of two really important brain chemicals,
Uh-huh.
dopamine,
Okay.
which we often hear about in relation to pleasure,
Right.
and acetylcholine, which is known to influence how dopamine works.
So think of these brain chemicals as messengers.
Okay.
Dopamine is often linked to that feeling of reward and drive, while acetylcholine has a more regulating role,
Right.
affecting the activity of other brain cells, including those that release dopamine.
And to actually see these messengers in action,
Okay.
they used a very thin fiber made of glass that could shine a light on the sensors.
Oh, wow.
And this allowed the researchers to track in real time when and how much of these chemicals were being released
Right.
as the mice engaged in sexual behavior.
So it's like having a tiny, incredibly sensitive light watching the brain's activity.
Right.
Yeah.
What's also important is that they didn't just watch the live behavior.
Right.
The researchers also did experiments on slices of brain tissue in the lab.
Okay.
Think of it like looking at the individual components up close
Yeah.
to really understand how they interact.
Right.
They also use genetic tools to change specific brain cells and their receptors,
Okay.
allowing them to really figure out the roles of different parts.
And to put all this information together, they used computer modeling.
And this helped them to understand the complex ways that acetylcholine and dopamine were interacting.
Yeah.
It's like building a virtual version of the system to see how all the pieces fit together
Right.
and influence each other.
That computer modeling part is key for making sense of all the information they gathered.
Yeah.
It lets them test their ideas and understand the fundamental rules that govern the brain chemistry they observed.
Okay, so with all those ways of looking at the brain in action,
Yes.
what did they actually discover?
Okay.
What were the main things they learned from watching the brains of these mice during sexual behavior?
One of the first things they noticed was quite surprising.
Okay.
They found that acetylcholine was released in a rhythmic way in the nucleus accumbens even before the mice started mounting.
Wow.
And then about 6 seconds later, dopamine started to be released.
Okay.
This suggests that acetylcholine might be preparing the brain for the upcoming dopamine activity, like getting things ready.
That's really interesting. Almost like the brain is warming up the engine before the car even starts moving.
Right.
And what happened in the brain during the actual act of insertion.
During insertion, the researchers saw that both acetylcholine and dopamine were released in rhythmic waves.
Okay.
And what's really fascinating is that these waves happen at the same pace as the mouse's thrusting movements.
Oh, wow.
About 1 or 2 times every second.
Okay.
It's like the brain chemistry is perfectly in sync with the physical actions.
That's incredible coordination.
It is.
And what about the mice that actually reached ejaculation?
Yeah.
Was there a different pattern of brain activity in those cases?
Yes, there was a very clear difference.
Okay.
Right before ejaculation occurred, the release of dopamine slowed down significantly.
Okay.
And then during the moment of ejaculation itself, there was a sudden rush of dopamine.
Oh, wow.
This suggests that this temporary dip, followed by a sharp increase in dopamine, is a key chemical signal for ejaculation.
So it's not just a steady climb.
Yeah.
In pleasure chemicals, there's this interesting
pattern.
rise and fall.
Yeah.
Right at that crucial point.
Exactly.
And they didn't just observe these patterns, right?
No.
They also looked at what happened when they directly influenced the dopamine system.
They were able to selectively activate different types of dopamine receptors.
Oh.
Think of these as different kinds of docking stations on brain cells that dopamine can bind to.
Okay.
They found that if they activated one type called D1R during insertion, the mice would go back to just mounting behavior. It interrupted the normal sequence of actions.
Almost like hitting the pause and rewind button on the sexual behavior.
Precisely.
Wow.
And if they activated another type of dopamine receptor called D2R, it completely stopped all sexual activity.
Oh, wow.
This really highlights how precisely dopamine needs to be regulated for the behavior to progress normally.
Right.
It's not just about having more or less dopamine.
Right.
It's about where and how it's acting.
So these different dopamine receptors seem to have very distinct and almost opposite roles in this whole process.
Yeah.
And they also identified a specific part of the nucleus accumbens that seemed particularly important.
Yes. They identified the ventral shell of the nucleus accumbens, or vsNAc for short, as a particularly critical area.
Okay.
This specific subregion showed these dual rhythms of acetylcholine and dopamine most strongly.
Got it.
It seems to be a key control center for these sequential behaviors.
And they followed up with those experiments on brain slices.
Yes.
To really get into the details of how acetylcholine and dopamine were interacting.
They did.
In this specific area.
Absolutely. These experiments showed that when they activated the brain cells that release acetylcholine in that ventral shell of the nucleus accumbens, it caused an increase in the release of dopamine.
Okay.
And they also found that this effect was influenced by different kinds of acetylcholine receptors.
So acetylcholine isn't just sitting there.
No.
It's actively influencing the release of dopamine.
Right. In this really important brain region.
Exactly.
And it sounds like dopamine was also having an effect back on the acetylcholine system.
Exactly. They discovered that the dopamine signals themselves regulate the activity of those acetylcholine-releasing brain cells in the ventral shell of the nucleus accumbens. And again, this regulation involved both D1 and D2 receptors.
Right.
Really showing this back and forth communication between the two brain chemical systems.
See, it's like a very delicate feedback loop where acetylcholine affects dopamine.
Right.
And then dopamine in turn affects acetylcholine.
Okay.
And the computer modeling helped confirm this idea of a reciprocal relationship.
Yes. The modeling strongly supported the idea that this two-way regulation between acetylcholine and dopamine through specific receptors, nicotinic acetylcholine receptors and D2 dopamine receptors, is really essential for creating those rhythmic patterns they observed.
And to really prove how important these specific components were.
They also used genetic tools to see what would happen if they reduced their levels, right?
That's right. When they genetically reduced the amount of D2 receptors, or the enzyme needed to produce acetylcholine, specifically in the ventral shell of the nucleus accumbens, they saw significant problems with both insertion and ejaculation. This gave strong evidence that these components are indeed necessary for these behaviors to happen normally.
So interfering with either acetylcholine or those specific dopamine receptors really disrupts the normal flow of sexual behavior.
Yes.
And they even use light to directly control the activity of these brain cells.
Yes, using a technique called optogenetics. They could use light to precisely control the activity of specific neurons. They found that if they artificially increased dopamine release in the ventral shell of the nucleus accumbens, it actually made the period of insertion last longer.
So more dopamine in that specific area seemed to prolong that particular stage?
It did.
And what happened when they reduced the activity?
Interestingly, they found that during insertion, the nerve cells expressing both D1 and D2 receptors were less active than usual in the ventral shell. And when they artificially activated D1 receptor neurons during insertion using light, the mice went back to repetitive mounting. Similar to what happened when they used drugs to activate those receptors. Activating D2 receptor neurons with light, on the other hand, immediately stopped all sexual behavior.
Again, really highlighting those distinct roles of the different dopamine receptor types.
Very much so.
And finally, what happened when they directly manipulated the acetylcholine neurons using light?
When they artificially activated the acetylcholine neurons in the ventral shell of the nucleus accumbens during insertion with light, it triggered rapid ejaculation.
Oh, wow.
And this was accompanied by a slowing down of the dopamine rhythm, exactly mirroring what they had observed naturally, just before ejaculation in the mice that successfully reached that stage.
So it really seems like acetylcholine plays a crucial role in that final switch to ejaculation.
It does.
Possibly by influencing that slowdown in dopamine. This is all incredibly interconnected.
It truly is. And understanding these precise brain chemical dynamics opens up some very promising possibilities for treating sexual difficulties.
Especially when we consider that initial statistic we talked about. The number of men who experience premature ejaculation. It sounds like this research might have direct implications there.
Absolutely. The finding that the balance between acetylcholine and dopamine in the nucleus accumbens seems to control how sexual behavior progresses could be particularly relevant for understanding and treating premature ejaculation. This study gives us a much more detailed picture of how dopamine works during sex and ejaculation, potentially leading to new medical treatments that target these specific brain chemical mechanisms.
And while this study was done in mice, the researchers do point out that the brain regions and brain chemical systems involved in sexual function might be similar in humans, which suggests that these findings could have broader relevance.
That's an important point to keep in mind. While we can't directly say that what happens in mice is exactly what happens in humans, the basic brain chemistry often works in similar ways across different species. This research gives us a foundation for future research in humans.
Of course, like with any scientific research, there are limitations to consider.
Yes, there are.
What are some of the main things we should keep in mind when thinking about these findings?
Firstly, as you mentioned, the study was done using male mice. Sexual behavior in females as well as in humans is much more complex and involves additional psychological, social, and hormonal factors that weren't part of the study. So these findings might not directly apply to those situations.
That makes sense.
Human
sexuality is definitely influenced by many different things.
It is.
And even between mice and humans, the actual physical behaviors are different,
Exactly. While the underlying brain regions and brain chemicals might be similar, the specific physical acts of sexual activity are quite different. between mice and humans.
Yeah.
We need to remember this when thinking about how these findings might relate to humans.
And were there any limitations in the methods used in the study itself?
Yes. The researchers noted that their attempt to reduce dopamine signaling in the ventral shell of the nucleus accumbens using light-based techniques wasn't completely effective.
Okay.
This means that some of the conclusions they could draw from those specific experiments might be limited.
So even with very advanced tools, there are always limitations in how precisely we can control biological systems. And what about the computer model they use? Is that a perfect representation of everything happening in the brain?
While the computer model gave valuable insights into how acetylcholine and dopamine might interact, it is by its nature a simplified version of a very complex biological system. It mainly focused on specific types of receptors. And the researchers acknowledged that other receptors, like muscarinic acetylcholine receptors, as well as other types of brain cells, likely play a role and need more investigation in the future.
So this study is a significant step forward.
It is.
But definitely not the final answer. It opens up a lot of new avenues for future research.
It does.
Now, it's always helpful to hear directly from the scientists involved.
Yes.
What did the lead researcher, Qinghua Liu, say about the findings?
Qinghua Liu emphasized the complexity of sexual behavior, stating, "Sexual behavior is a complex sequence of events." The study revealed the dynamics of how different chemicals work together in the brain to regulate the transitions through different stages of male sexual behavior. This really highlights the study's focus on the changing roles of these brain chemicals.
And what about the perspective of the first author of the paper, Aimi Asaka?
Aimi Asaka focused on the level of detail they were able to achieve, noting, "We are able to look at these events at a very fine time resolution to understand how brain chemicals interact with each other. Now we have a precise understanding of how dopamine works during sex and ejaculation. So I believe our study has opened the door to the development of clinical treatments. This really underscores the potential for future medical applications."
And it sounds like the researchers are particularly hopeful that their findings could lead to new ways of treating sexual difficulties.
They are.
Especially premature ejaculation.
That's a key potential outcome. By precisely mapping the brain chemical events that lead to ejaculation. The study provides possible targets for treatments aimed at addressing premature ejaculation.
This has been a truly deep dive into the brain chemistry of a very fundamental behavior. It really makes you think about how much of what we do, even things that feel like instinct, are actually rooted in these incredibly fine-tuned balances of brain chemicals.
This leads to an interesting thought for you, our listener. Consider how deeply our behaviors, even those that feel primal, are connected to this intricate dance of brain chemistry. This study gives us a glimpse into that fundamental level of control. What other complex human experiences might be governed by similar intricate ballets of brain chemicals?
That's a really thought-provoking question.
It is.
And if this exploration into the brain chemistry of sexual behavior has sparked your curiosity, we encourage you to look into the broader research on the roles of dopamine and acetylcholine in things like reward motivation and even how we move. Understanding these basic mechanisms is really key to unlocking the bigger mysteries of the brain and developing future treatments for a whole range of conditions.
What's remarkable is that by understanding these fundamental building blocks of brain activity, we can gain insights into not just specific behaviors, but also the underlying principles that govern a wide variety of human experiences.
Thanks for taking this deep dive with us today.
A pleasure.
The key takeaway is the incredible precision and rhythmic interplay of acetylcholine and dopamine in a specific brain region, the ventral shell of the nucleus accumbens. And how this controls the progression of male sexual behavior with important implications for understanding and potentially treating sexual dysfunctions.
If we step back and look at the bigger picture, this research really highlights the power of combining advanced neuroscience techniques with computer modeling to unravel the complexities of the brain.
And as research continues to uncover these intricate brain chemical processes, we move closer and closer to a deeper understanding of ourselves.
Absolutely.
And new possibilities for addressing some of the challenges we face as humans.
That's the exciting part.
It is.
Thanks for tuning in to the Health Headliner podcast by To Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.